So, one of our first special senses, so again, has a specific organ, the tongue. And there are 5 submodalities to taste, right? So subgroups: sour, salty, bitter, sweet, umami. Um, we may have something. There's always speculation because there's no way you could test every human being. So the data we have are based on very limited number of human beings. So we can agree upon this, though, right? Sour, sweet, bitter, sour, salty, bitter, sweet, and umami, which is savory, right? So here we are looking at the tongue. The tongue, if you look at it closely, is a whole bunch of bumps, right? And the bumps are actually different sizes. You may not have noticed that, but if you have, you are correct. So we do talk about these, right? Fungiform, filiform, and foliate. And I'm not going to make you discriminate between those different papillae, but it's on these papillae in which we find the taste buds. So a taste bud really looks like a little flower bud. They're really cool to see, and we'll look at these in the lab. Here's kind of one right here. They have to have a right contact with the actual sensation. Thank you, sir. And to do that, they have to open to the surface of the tongue. And so what happens is that saliva mixes right with the chemicals in your food, the molecules that make up your food. And then the saliva, now in a dissolved form, the saliva washes over and down across all these papillae and makes contact with these sensory cells, these receptors, all right? So, which they just call the gustatory cell. So, and it turns out that these gustatory cells have these hairs. So this is the sensory part, right? It's making contact through this opening called the taste pore. And then there's these support cells around this. But these are the true sensors, all right? So when we look at the tongue itself, it is a bunch of stratified squamous. Yeah, don't forget those things, right? The surface, this helps to protect your mouth. Most of your oral cavity is stratified squamous. There are 4 types, which contain taste buds, 4 types of these actual papillae. They have gustatory receptor cells, right? Because here's a circumvallate. Sorry, I forgot to add that one. So 1, 2, 3, 4. And they have gustatory receptor cells, which are shown here, right? And what happens is we activate neurons and sense. So these connect. You see the leading away from the surface of the tongue. And we're going to then travel in bundles through the facial nerve, which is mixed, glossopharyngeal, and vagal, which are mixed as well. And so this is carrying, again, this sensory information to the taste cortex, which is kind of scattered around a lot in the insula and some in the parietal lobe, from what we understand. All right? So here we talk about how this works. So salty is a perception of sodium ions in saliva. This causes depolarization, again, of these, what they're now calling here the taste cells, what we call the gustatory cells in the last one. Sour is a perception of the hydrogen ions, acids, right? So acids have a sour taste, which enter and cause depolarization, right? So again, different receptor cells have sensitivity to different things. That's why we're talking about these submodalities. Sweet is the presence of glucose and other molecules, and this goes through, I'm just telling you, through a G protein. You don't have to know that. Bitter are alkaloids, so oftentimes these are bases, right? Alkaline things can cause this depolarization, and also there can be hyperpolarization from bitter. But let's, again, we're in G protein changes, we're going to just focus on, again, alkaloids being part of the bitter, and this depolarizes, starts the action potential. Umami is the presence of glutamate, and it also works through G protein signaling, as opposed to just outright depolarization. Again, I'm not going to ask you about what happens once it binds. We're just going to assume we're just going to generally say an action potential is initiated. But it is initiated through different mechanisms, is all that's being said here. All right? So these dissolved molecules or ions, right? Amino acids and hydrogen ions, sodium atoms, bind receptors on the hair. This initiates action potentials. That's what we want you to get. Carried by sensory neurons to the parietal lobe and also the insular lobe, though, this OpenStax doesn't talk a lot about the insula. Sensory adaptation is similar to smell. So we haven't mentioned this yet, but adaptation is this idea that you stop being able to perceive the sense. And so smell and taste do this, right? And please consider. So if you first put something that tastes really bitter or hot or gives you a sensation of heat, right? Strong. After a while, you lose that intense perception of the sense. And that's because of adaptation. Adaptation mechanistically is the idea that the receptors, right, those gustatory cells, stop being able to be stimulated as frequently, and they need to reset, but it takes longer to reset or repolarize, and as such, you cannot, right, perceive the same way, in the same intensity that you had in the beginning, right? That's called adaptation. You're becoming used to it, right? So taste cells also undergo wear and tear. They are epithelial in nature, right? So they're modified neural cells. And they divide. So you can replace these in about 3 days, and that's good. So you can maintain your taste sensation through your life. Though it does diminish to some extent because the replacement time period gets longer as we age. All right? This is for your information, so you would not need to know this on a test, the pathways. So the anterior 1/3 of your tongue is carried by the facial nerve, the posterior glossopharyngeal, and the vagus is a root. Axons of these three different nerves, right? Synapse in the gustatory region of the brain stem nuclei. Again, this is for your information. And then they synapse in the thalamus. As we said, a lot of sensory passes through the thalamus. And then the thalamus projects to parietal lobe and insula. All right? Olfaction. So olfaction, another special sense. Smell, right, is right here in the nasal cavity. So a chance for encounter. And so as you inhale air, you inhale molecules that are being carried, right, diffusing through the air, right? Remember, that's the way it works. And so here we're looking at a cartoon of this epithelium, all right? So here what we have, again, is we have these, right, contact made through these cilia that are projecting through, right? And here we've got some mucus surrounding them as protection, but here they are down here. So here's the cilia. And so smell, right, this is the opening of the cavity. So again, opportunity to contact, opportunity to start, and action potential. These are actually types of bipolar neurons, right? You can see there's the cell body, and there's two and only two processes. Dendrites, axons, okay? And here they are synapsing. They move through, right, the ethmoid bone. There's these little cribriform, right, little tiny foramina openings, and so the neurons move through here, all right? And then they synapse, and we're going to pick this up with the olfactory, right, nerve, which then becomes the olfactory tract as it moves in. Okay, so here again, inhaling, here's our pathway, small region in the nasal cavity, and it's made up of this olfactory epithelium. Its smell happens in response to, right, stimulating, exciting these olfactory receptor cells, which are bipolar neurons, as we said. The odorants, again, dissolve, just like the chemicals in your food dissolve in saliva, the odorants dissolve in mucus, and they bind to, right, these cilia, these parts of the cell. And so if they bind in the right combination, you get it excited, and an action potential starts and is moved, right? This excitation is moved. So axons from the olfactory neurons form the olfactory nerve, which is cranial nerve 1, and they pass through the cribriform plate of the ethmoid bone. Don't have to know that word, right? To enter the olfactory bulb, all right? They synapse with interneurons, which take these to the olfactory cortex, because remember, olfaction is one that doesn't actually pass through the thalamus. And so inferior medial areas of the temporal lobe go there, as well as the limbic system, which is a combination of the cerebrum and the diencephalon, and the hypothalamus, especially the mammillary bodies, and smell becomes associated again with memory and emotions. All right, hearing. So audition, right? Auditory, hearing. Here again, here's our temporal bone. So this is our external acoustic meatus right here. So organs, we have external, we have middle, and then we have internal. And the internal are the truly sensory. The external and middle are just transmitting, right, the change, sound energy, into the inner. And they are modified to do this, right, to gather sound waves, right? Your auricle is this fleshy, elastic cartilage that you think of as your ear. And we move through, yes, the ear canal, right, or auditory canal, into the, right, tympanic membrane. That's our eardrum, and you need to call it the tympanic membrane, please. And we see this on the models, right? The middle ear is an air-filled chamber, so it is a small chamber and must be continuous with the oral cavity through this eustachian tube or auditory tube. All right, and so this would end in, this is when we talk about middle ear infections, it's bacteria, viruses colonizing and moving up from the oral cavity into the middle ear, because it is open, right? And we need this opening to equalize pressure in this small when we change in altitude, right? Or a change in going underwater, right? You have to have the ability to open up, and that's why we swallow, chew gum to equalize pressure in here with the atmospheric pressure. All right, so the inner ear, right? And we'll look at these ossicles, these three bones, the malleus, the incus, and the stapes. And then the inner ear is a bunch of fluid-filled, right, bony and membranous chambers. And so we'll have this structure called the cochlea, we'll have a structure called the vestibule, and then we'll have these, yes, they look at semicircular canals, and they will all be part of the inner ear. So the external and middle ear are really related to hearing. The inner ear gives us hearing here at the cochlea and balance. Okay, and here's the nerves that carry the action potentials that really begin here. And hearing and balance are both mechanoreceptors, right? Smell and taste were chemoreceptors. All right, so here, the external ear, again, the auricle is a fleshy part outside the head, opens to move through this, they call it the ear canal, but the external acoustic meatus is the bone, and we do call this, and it's still an external acoustic meatus, leads to a tympanic membrane, so the auricle collects and directs sound. In other mammals, we can move, right? They can adjust their ears to collect that sound. We're not so good at doing that anymore. We kind of made some trade-offs here, but you're aware that that's, again, an ability for us to capture the stimuli. And it's very important to hear, the subtle sounds. So your ear is really designed to maximize. There is protective features for intense sound, but your ear is really there to manage, right, really quiet sounds, right? Because that's how we know that there's prey around or a predator around. So we have to consider why we evolved these different senses, and this one's a pretty obvious one. All right? And smell is the same way, honestly, and so is sight. So hearing, smell, and sight are very much a part of our survival. So this meatus, this tunnel or canal, transmits the sound energy. It's lined with hairs and ceruminous glands. That's where you get the earwax. And this helps to trap anything, again, because you could colonize, right? This is an open area to the outside world, and we want to be protective. So these are protective things. The eardrum or tympanic membrane, which is what we need to call it, right, is a thin layer of connective tissue between epithelium and vibrates. That's the important thing. It moves in response to these sound energies, which is traveling in waves coming in. All right? So the middle ear here is actually then also going to be responding to the energy and trying to transmit it. So the auditory ossicles, the malleus, the incus, and the stapes, also known as the hammer and the anvil and the stirrups, but we need to call it malleus, mallet, hammer, incus, right, anvil, and stapes are connected to each other, all right? And they're attached then to this temporal bone through ligaments, and we are transmitting vibrations from this membrane through the bone, right, to this membrane that's covered here called the oval window, all right? So transmission from the outside world through the middle ear and in. Now this situation where we have a big surface area and then a much smaller really amplifies sound. All right, and that's what we said. We're after. We really are often more in need of hearing the really, really soft, or what we consider quiet, low energy sounds.